Another example of a enzyme that utilizes multiple types of catalysis is chymotrypsin. Chymotrypsin is very important. It's used during digestion. And what it does is it actually breaks down proteins. So it's what's called a protease. The ace part here again just means that we are breaking or cutting, and prote is for the protein. So it takes big proteins and breaks them down into small peptides. Why does it do this? Of course, we want to consume proteins. And by consuming proteins, we can get small amino acids and small peptides that we can then utilize to build other things in our body. So this is doing the job of breaking down proteins that are coming in from our diet so that we can use them as building blocks for our own proteins and our own enzymes. So chymotrypsin is actually one of several types of proteases. And I suggest you look into these proteases. There are different types. There's things like just regular trypsin, again, chymotrypsin. And all of these proteases cut at a very specific location along a peptide backbone. So they will only pick to cut at a specific junction between amino acids. This protease, in this example, cleaves the peptide bond that is adjacent to any aromatic amino acid. So chymotrypsin, it is able to recognize a peptide and a protein, and it only can bind onto a site that is next to an aromatic amino acid. And that is where it will catalyze the reaction and cleave. And again, there are different proteases that recognize different parts of proteins, and they will chop the proteins at those different junctions. So for example, here we have a tyrosine, and you can see that the chymotrypsin is going to cleave at the peptide bond that is next to, adjacent to the aromatic amino acid. And remember, we always read from this amino terminal to carboxyl terminal. So it's, when reading correctly, it's going to be the peptide bond next over to the right of the aromatic amino acid for chymotrypsin. Now, chymotrypsin is used as an example a lot when talking about enzymatic mechanisms because it uses a lot of them. We talked about different types of enzymatic mechanisms, and chymotrypsin, throughout its process of catalysis, utilizes a bunch of different mechanisms together to catalyze this one reaction. So here you can see chymotrypsin utilizes these amino acids within its binding pocket. It has a hydrophobic pocket and then another area that is hydrophilic. The hydrophobic pocket of the enzyme, this little part of it that is hydrophobic within the active site binding area, is what helps bind that aromatic ring. That is how chymotrypsin is specific for looking for these aromatic amino acids, because those aromatic amino acids are going to be attracted to the hydrophobic pocket, and that's kind of how it situates the peptide into that region to then allow for the catalysis. Now, chymotrypsin works through what is called a triad of amino acids. It's just 3 amino acids that are doing the bulk of the work during the process of its catalysis. And that is a glycine, a serine, a histidine, this glycine, a serine, a histidine, and a aspartate that are the main triad of amino acids. Though glycine is part of the reaction in its stabilization as well. Normally, though, this aspartate, histidine, and serine are the catalytic triad that is mentioned generally. So here you can just see the first step of the chymotrypsin chymotrypsin mechanism is to bind the substrate. Again, this is any polypeptide, any protein, and it's going to bind next to an aromatic amino acid. So the aromatic amino acid is attracted to that hydrophobic pocket. And then it is stabilized in position utilizing hydrogen bonds through these other amino acids in the pocket. You can see that serine, for example, makes a hydrogen bond with a carbonyl here, and the histidine makes a hydrogen bond and then actually donates its hydrogen, but at first is stabilizing its hydrogen in bonding to help stabilize this peptide. Now, again, I'm not going to get too far into the mechanism here, but I want you to see, again, the various types of catalysis that are happening. So chymotrypsin is utilizing a nucleophilic attack. So again, this is actually forming a short-lived intermediate of covalent bonding. It also utilizes hydrogen acid base or a proton transfer using acid base catalysis as well. And again, I want to clarify here that in the end of this mechanism, water is utilized, and this is very common, to essentially regenerate the catalyst in the completion of the product formation. So you can see, for example, water is added here to regenerate the proton that was on this original histidine. And by doing this, it is also helping to remove the final peptide piece as well.